All right. Good afternoon and welcome to the aquatic section.
Don't forget to take notes if it's interesting to you.
It's all going to be interesting to them.
Absolutely.
Our first speaker for today will be Kim Sullivan, who is down at the E-Hunty, and followed by Molly Allen with the Northern Rhode Island Conservation District. And wrapping it up is Maureen Dwyer from Narragansett Bay. We have Coventry High School on the line, and we have Natalie from East Providence High School. So if you ladies could take it away, that'd be great. Thank you very much.
All right. So I'm going to go ahead and start off. I know we all have a tendency to love our topic of water, so we're not going to we're going to start off by sharing and get right into the presentation. Water quality. Let's talk a little bit about, we're just going to go over the basics. So it's been a couple of years since we've gone over the basics. We kind of tightened our presentations up a little bit, me on my end. So we're going to, let's talk a little bit about water, if I can get my, all right. So everyone can see my screen, right?
Yes.
All right,
Yes.
excellent. When we're talking about Earth's water, and you look at the globe, pretty much you're about 70% of Earth is water. And of that water, about 97% is actually oceanic water. So I found this graphic. I really kind of liked it because it really gave a good depiction of what's what. So with 97% of our water being ocean and very saline, we can't drink it. It's not potable. So the 3% is fresh water. Problem with fresh water is that most of that fresh water is all wrapped up in our glaciers, as well as underneath us. So for accessible water, or stuff that we would actually use for drinking and potable uses, swimming, recreation, is basically only about 1%, or a little bit less than 1% of all of the Earth's water. So I feel like this representation kind of gives you that idea, and where that fresh water is. 52% are lakes, about 1% is rivers. There's water vapor, the clouds, in that whole water cycle, which is something you definitely want to study, review, and really kind of take into account what that water cycle is. And then soil moisture. I'm sure Mallory will talk a little bit about aquifers and how it percolates in. So I'm not going to take any of that and head that way. So we're talking what's going on with our waters. So with all that, that 3% of fresh water, what's actually going on in some of the issues that we're dealing with. And we're actually still dealing with issues from hundreds of years ago. From Mill, you'll see that Slater Mill is right over here on the left corner of your screen. And not only did they erect a whole bunch of dams, which stopped connectivity of fish connectivity, but it also, they were dumping heavy metals and stuff into the water. So a lot of our waters here in Rhode Island are dammed up. There are 519 dams as of by 1870. And so not only are they dammed up and affected the fisheries, but they also have effluent in it, such as the PCBs that came from the mills. The other thing that we're combating over on the right-hand side are invasive species. This is a picture of the snakehead, but from anywhere from plants, aquatics, to actual invasive species. Carp has been proliferating in our waters and isn't native. So we're looking at problems with invasive species, especially as climate change changes our waters are warming up, and it's allowing these other species to come on up into not only our saltwater basins, but also our freshwater. The other thing are man-made chemicals. So y'all heard of Teflon, that's the coating in the pans and stuff from DuPont. So PFAS and PFAS, these are man-made chemicals that actually don't break down or take a very, very long time to break down. And that's not even including the plastics that we're starting to deal with as well. And of course, climate change. So to the right, you'll see a pond that is actually experiencing a cyanobacteria or a toxic algal bloom that is actually a source. Those toxins can create neural problems within humans, as well as it can actually kill your dogs if you were to allow them to drink that water. So the good news is that there are some federal policies in place that I would like you guys to know about. For healthy habitats, the Clean Water Act came out in 1972, and basically it was designed to regulate pollutants in our waters. And basically it's kind of an amendment to an earlier time of 1948, when they started really kind of getting into those clean water, anti-pollution, realizing that what we're putting into the water wasn't so great for our ecosystem. And then on top of that, we extended it to safe drinking water. By right, we should be allowed to have safe drinking water. So the CDC tests over 90 contaminants for your drinking water, which is kind of a good thing. But, you know, there's some contaminants that they still can't account for yet. They don't necessarily know how to adequately test for the PFAS or the plastics or anything like that. So those are tests that are starting to develop as we're going through the progressing through time. So it comes down to, how do we know if we have good water? And this is something, you know, all three of us are very passionate about, is we will, you know, how do we carry on these good stewardship practices if we don't know what we're looking for, or how to even define good water? So the EPA actually has a couple of good ways of testing water and stuff, and came across something over the past couple months that, you know, focusing on what, you know, just looking at the water system, smelling the water system, what we can actually as humans kind of determine, not necessarily by sight, but, you know, seeing what's there, and by observations, if it has a funky smell, you're probably not going to drink it, that type. So in prior talks about water quality with the Envirothon, I had not touched on the importance of, you know, just using your senses and seeing what's going on and observing. And I think that's something that, you know, as scientists, you can start learning with all four disciplines, is that sensory observation. And of course, we have water chemistry testing. So some things I'm going to be going into in the next couple slides are what parameters we use for that water chemistry testing, including temperature, oxygen, pH, salinity, turbidity, and nutrients such as nitrogen and phosphorus. And finally, one of my favorite topics, and something that you guys will be expected to know on the day of the test, is biological monitoring, using the bugs to actually tell you whether or not it's good or bad water quality. All right, I'm going to pause here. Are there any questions at all yet? All right. So let's go into some water chemistry testing. One of the most basic things as water people that we go and talk about is actually temperature. Temperature actually, you know, it's basically the measure of heat energy held in a single molecule of water. And basically, it helps us determine whether or not it's going to have higher levels of oxygen, lower levels of oxygen. So generally speaking, water temperature, lower water temperature usually means higher oxygen levels, which we all need oxygen, including our fish, in order to survive. And then you know, lower temperatures often mean that less oxygen is held in that water. And it actually determines density, water density. Now, do you guys have a on the right-hand side, I see the thermometer on my screen, but I'm not seeing the water temperature thermal stratification. Are you guys seeing that?
No, it's blank.
All right, so I'm actually going to stop sharing and reshare over here. I'm not sure why it's not coming through, but I did want to show you this right over here, the water temperature stratification. So what happens is as the water gets colder, it gets denser, starts falling down to the bottom of the water system, especially in our lakes and ponds. And it actually creates a stratification or actually barrier of the flow of water. So that becomes severely important when we're talking about springtime and fall time turnover of those water systems. So at that denser temperature, that colder temperature, there's no oxygen that's actually penetrating. There's no light that's penetrating, and there's no circulation. So that's kind of a very oxygen depleted area. So what happens is during the summer, that whole system, or during spring, that whole system, as the ice is melting, and ice starts falling down, it actually brings up that deoxygenated water, the anaerobic water, and basically that's what ends up going for commonly fish kills, so on and so forth. So that water stratification is pretty important. I'm just going to keep using the slide here because I feel that if that didn't work, my other slide won't work. So the second parameter that we test through water chemistry is oxygen. So as again, we all require oxygen in order to live. That includes the little organisms in the water, as well as the plants and stuff. So anyway, the higher the dissolved oxygen, you're going to find that your fish populations are going to thrive. So generally speaking, right around anything under 4 parts per million, fish are not able to survive, and that's just because they're not able to handle those lower dissolved oxygen levels. And related to the graphic on the bottom, it just kind of shows you the more oxygen that's held in the water, usually colder running water holds the most oxygen, that you're going to have greater diversity and greater plentiful fish in that area. Generally speaking, there are exceptions. And we have different tools for testing oxygen. That includes water testing kits that you see up here in the upper left, and the YSI meter up on the middle of the screen. And that is an actual DO meter that will generate without having to using testing or titration. The next one we want to talk about is pH. So pH is a logarithm of hydrogen oxygen or hydrogen ions within the water. And so the greater the pH, the more basic it is, the lower the pH, the more acidic. And I like seeing this kind of graphic for the pH scale because it kind of gives you real life examples of what we're looking for. Lye would be like 14, hydrochloric acid would be a 1 on that scale. And usually fish itself, they can handle anything for around 6, depending on the species, anywhere from 6 to a 9 on that pH scale. And there are different ways to test for it. On the right-hand side, you'll see there's testing strips, titration kits, as well as meters. All right, another thing that we test for is salinity, especially here in Rhode Island. We have a very unique ecosystem where we definitely have saltwater and freshwater. And if anyone ever fishes in the Narrow River, there are times that you can catch a striped bass, which is a saltwater fish, and a trout, which is a freshwater fish. And that all becomes this kind of gradient that you see here on the left and the bottom, showing how the denser saltwater kind of goes underneath in the freshwater right before it mixes. So depending on the river flowing out over the bay, so the freshwater ends up being a little bit on top, allowing those saltwater organisms such as crabs and saltwater fish to stay in that lower edge. So you can actually find crabs and striped bass and shad in our narrow river because that interface is so long. And this is a hydrometer, and that is how we test the. We can also use a refractometer. That is a more expensive piece of equipment to determine what the salinity is. A hydrometer is usually quick and easy. Another thing that determines turbidity, turbidity actually affects the soils, the suspended solids in the water, actually affects how much light can penetrate into a body of water. Now, clear water doesn't necessarily mean that it's perfectly great water. Up in the Great Lakes, there's been instances of invasive mussels that have actually cleared the water and taken the nutrients out of the water so much that it's super clear, but not a whole heck of a lot of life can live in there because of there's just no nutrients in the water. That those mussels have done too much to filter that all out. But the Secchi disc is one of the most common most common tools for turbidity. And pretty much depending on the clearness or the murkiness of the water it's given, it kind of gives you an idea of what you're looking for. So the other thing that I talked about are limiting nutrients. I mentioned that the mussels up in the Great Lakes have eliminated definitely some of those nutrients. But too much of a good, too much nutrients is too much of a good thing. Comes to eutrophication in our freshwater system. So we call them limiting factors. So phosphorus would be a limiting factor for our freshwater systems. Too much will mean that there's a huge amount of algal blooms. And then what happens is that when those the vegetation blooms and then dies, it actually sucks out the nutrients or the oxygen in the water as those plants are decomposing. So what happens is generally no lower oxygen, we do end up with fish kills. Nitrogen on the other side is a limiting factor for our saltwater systems. And E. coli is also a harmful bacteria that we do test for. All right, and now we're launching into one of my favorite parts of water quality, and that is macroinvertebrates or biological monitoring. So based on the insects that are in the water, you can actually determine what kind of water quality it is. And so we call them macroinvertebrates. So what is a macroinvertebrate? So basically it's an organism without a backbone that you can see with your naked eye, visible. It's macro, unlike a micro, like microscopes, where you need a lens to make that image bigger. So it's completely opposite of a micro or microbe. So often they're the nymphal stages of terrestrial critters, such as mosquito larvae, mosquitoes, stoneflies, mayflies, and this here is your dragonflies. So you can actually see the dragonfly head right here. It matches kind of the dragonfly head there. This would be the wing cases that when they emerge, come on out, and actually you can see the tail right there, and it's similar there. But you probably wouldn't If you saw this creature coming out of the water, you would not think that it was a dragonfly larva. So they've become water quality indicators because they have spent years in the water. We're talking 2-3 years, depending on the species. So those that spend the longer time in the water require more oxygen, more nutrients, more better water quality overall in order for their survival. Dragonflies spend about 2 to 3 years in that water system. So they do require some pretty good oxygen. And EPA has a written protocol for that monitoring. It's very scientific, so that it can be replicated over and over during different times of the year. When I do the habitat assessments, and what we will be doing at the Envirothon training will be a quick and dirty. Get in there with your waders on, with your net, and just let's grab and see what it is, and kind of make an overall assessment of that water quality of the water body we're testing for that. So when you start talking about parameters, so we have to have some sort of guidance, like what kind of organisms live in that good water quality, what kind of organisms can live in any kind of water quality, even though that water that isn't so great for swimming or fishing or anything else. And so they've actually formulated 3 groups. Some testing has it for, but here when we're doing the Envirothon, we're basing it off of 3 groups. So group 1, they are our mayflies. They are our organisms that require high oxygen. They're very pollution intolerant, which means, that turbidity comes into play. So pollution isn't just, runoff from, a toxic waste site. It's pollution, anything that's going to be an environmental stressor on that. Too much fertilizer going in, too much of siltation going in, too salinity rise. So everything that would consider as pollution, they are intolerant to that. They don't like changes in their ecosystem. Group 2, they're a little bit more resilient. They require a little less oxygen than group 1, but they still require some really good water quality in order to survive. Again, they last in our ecosystem quite a few years in order to live and stuff. And they are somewhat pollution intolerant. They can handle a little bit more influx than the group 1, but they still need to have that pollution barrier. And group 3 requires little oxygen, and they are pollution tolerant. Now, how am I doing with time? Do we need to switch over?
You've taken about half an hour.
Oh my gosh. Okay, let's switch. I'm going to end it here. I wish you had told me.
It's okay. It's all good info.
All right, there's the stop
I
share.
can make mine on the quicker side.
Okay.
Hi, everybody. My name's Molly. I'm from Northern Rhode Island Conservation District. Special hi to Situate Middle School, who I saw popped in. I'm thrilled to see you all here thinking about Envirothon or doing Envirothon. I don't know which. I'm just happy to see you here. All right. So as you probably noticed, the aquatics section is huge. They basically want you to have my grad degree level of aquatics knowledge. So we're touching all the bases. You're not going to remember everything we say, but our hope is that you're going to remember, hey, they were talking about the concept of macroinvertebrates. They were talking about the concept of watershed delineation. And you'll remember that that's something you have to go back to closer to when you're studying for the test. So as such, my presentation also includes homework. So we have a lot of past videos and past trainings online that you can check out. If you want to do some studying but want to keep it easy, pop on a video. We've got you. We have a groundwater model video that I made that ended up with I want to say 500 views on YouTube. Very exciting for me because who knows? But we're going to dive right into one skill that has been asked on the national test for the last few years, which is delineating watersheds. Delineating means looking at a map and seeing what the watershed is. So this is a topographic map. If anybody does not know what that is, stop me right here. Wait a second. But as it seems like most of you know, a topographic map is a map that shows how the elevation changes on the landscape. Now, when you're looking at a topographic map, when you see a little circle, you have found the top of a hill or the top of a mountain. So in this map here, they have put stars on all the high points of the hills and mountains. That is one of the steps of delineating a watershed. But step one is you want to identify the outlet point. So when we say the watershed of Narragansett Bay or the watershed of the Scituate Reservoir, we mean that that is all the land, all the hills and valleys that catch water and bring it down to that place. All the hills and land that will catch water and bring it to the Scituate Reservoir or to Narragansett Bay. So in this exercise here, which is a similar one was seen on the national test this year that we got to look at after the students from Wheeler took it, they were asked to delineate or outline the watershed of this water body right here, oopsies, which has little stripes on it. So they looked at the map around that water body. They found the high points of all of the hills and valleys, and they put stars on them. And then they connected the dots. They connected all the high points so that the lines, we're going to cross the lines on the topographic map at about a right angle. So they found the high points, connected them, and that's the outline of the watershed. So all of the rain or snow that would fall in this area right here would end up in that lake that they have highlighted on the bottom. This is a broad, quick outline to how we do this. If we have an in-person training, I might bring some worksheets where we can practice it in person. I also have a link in this PowerPoint to an activity sheet that you can use to practice this concept. I can't teach it to you in 15 minutes, but I want you to know that it exists. Start wrapping your mind around it, start thinking about it, because it has been showing up on the national tests pretty consistently, which is why we added it to our curriculum. Something else that's been showing up on the national tests a lot lately that I think personally is kind of a fun one is stream order. So stream order is a way to classify our streams based on their size and their connections to other streams. So a tributary stream, that's a good vocab word to know, tributary, is a small stream that leads into a bigger stream. This aerial photo, which I think is so cool, I actually don't know where it is from, but I love it, shows how little streams lead into big streams like this. So our very largest rivers might be a 12th order stream, but the very smallest streams on a map, the types of streams that those of you who live in suburban or rural Rhode Island might even have in your backyards, just those little ones that only run in the springtime, kind of dry up in the fall, those are first order streams. So how do you do it? Now, there are a couple of different methods to identify stream order. The one we use is the Strahler method. It was created by a hydrologist named Strahler. Basically, to do this, you assign every stream a number. So you find the smallest streams on your map, the ones on the end. These are first order streams. Now, last year's Envirothon, I showed a diagram very, very similar to this one. And a lot of students came up and asked me, which direction are the streams flowing? And I told them that they should be able to tell that from the picture. The smaller streams always flow into the bigger streams. The smaller streams always combine into larger streams, not the other way around. So if you see a picture like this, look at the ends. The water flows to the next intersection where it's going to combine with another stream. You don't need an arrow to tell you that. And now if you've watched this, you're going to get a free point on the test probably. So the very endpoint streams, those are first order streams. You number them number 1. When two first order streams meet, that stream becomes a second order stream. When two second order streams meet, they become a third order stream, and so on and so on. Whenever two streams of the same order meet, the resulting stream increases by 1 order. Now, if you look at the bottom of this picture, we have a small first order stream running into a larger stream. That doesn't change the stream order. The larger stream stays the same number until it meets another stream that is the same order that it is. Now, I found a great YouTube video that demonstrates this. It's only about 2 minutes long, so I'm going to pop it on for you. All right. 1 second, just arranging the share. Excuse me squinting at my screen. I don't know if you're going to be able to hear the audio, but you don't need to. I'll narrate.
I like to do these. I start with the first order streams.
Can people hear the
Now,
audio?
you know that these are the streams with
Yes.
no tributaries.
Okay.
They are higher elevation, usually spring fed. So that means they're going to be colder, they're faster moving, and they're going to flow downhill to the higher order streams. So let's take a look up at the left-hand corner here. I'm seeing a first order stream here and here. Now, when they meet, they're 2 streams of the same order. So the rule is when you have that occurring, they're going to create a stream of the next highest order. So it's going to be a second order stream right there. And this guy's going to flow to this point. We got a whole river system to the right there that we have to go to its beginnings to figure out what it is at that point. So over on the right-hand side, I got a first here, I got a first here. And when they meet at this point, it's going to create a second. Same sort of idea. That one's going to keep flowing. We got a first order stream here flowing in, but that's not going to change anything because when they meet, they are 2 different orders. So the stream of the higher order is going to take precedence and continue flowing as it is. So that second order is going to stay as a second. It's going to continue down to the point where it meets with the other second order, and it's going to create a third order stream. This guy's going to keep flowing down. We got a first order flowing in here, but that's not going to change anything. So the third order flows down to this point. Now we got to figure out this whole system to the bottom of the screen here and go to its beginnings to figure it out. So let's take a look at the bottom left. We got a first here. We got a first. When they meet, it's a second. To the bottom in the middle, we got a first order. We got a first order when they meet, same idea. Got a second there. So when these 2 seconds meet at this point, you know what's going to be? Third order. That third order is going to keep going. Got a first flowing in, no big deal. That third order is going to continue up to meet the other third order. So we're going to have a fourth order stream right there. Along the way, we got 2 first order streams flowing in, no big deal. And a little bit further down, we have another stream flowing into the fourth at this point. It's a little bit more complicated here, but again, just backtrack to its beginnings and we'll figure it out. So we got a first order here, we got a first order here. When they come together, we got a second order. So here we go, we got a second order stream flowing into a fourth, but since they're not of the same order, the one of the greater order will take precedence. So the fourth order is going to continue down as a fourth and flow into the pond. Thanks for watching.
Sometimes I just want to rent a car. Go, you know?
I do know.
Stop in the
And I think I
ads,
can-
stop in the ads. Great. So this is a skill that is easier if you practice it. So what you're going to want to do is get on Google stream order practice, and find worksheets that other students, teachers, professors have put online, and just practice it. You can even make up your own for your friends. It's just drawing rivers and how they get together. So we have a link to another video, and like I said, homework is to practice. So what's the point? Why are we doing the stream order? Is it just fun to label things with numbers? No. Stream order can tell you a lot about what the stream's characteristics are probably going to be. So first order streams are going to be more susceptible to stormwater or non-point pollution. Like they're less water, so a little pollution is going to make a bigger difference. Higher order rivers tend to move more slowly. They might collect more debris and pollution, might also be a bigger flooding risk. Stream order can help predict what types of life might be present in the river. So to connect that to the macroinvertebrates, a very clean first or second order stream might have higher, more sensitive critters living in it than a larger stream. It's helpful in map making to show the relative sizes of brooks, rivers, and streams. Overall, it's just one good tool hydrologists can use to understand a lot about our waterways. So we're going to do a quick review of aquifers and how they work. So aquifers is our underground water. Many people think our groundwater looks like this, like flowing underground rivers. That's not what it looks like in most cases, though, although in some places, which is why we have this picture. A lot of times it looks just like water seeping through the cracks of big pieces of granite, like we see in this picture here. That water freezes when it hits the air in the cold winter. I think this was from 295. It can also look like wet soil. When you look underground, you're going to have a zone of saturation. That's where the groundwater is. The soil's wet up to that point, and it can be moving. It can be flowing underground. In many cases, we drill wells into the groundwater to use for our drinking water. Scituate and Coventry both have high percentages of people that get their drinking water from wells that are located near their house, as opposed to reservoirs. A lot of Coventry does get water from the reservoir, and some of Scituate as well. Now, wells in your backyard, if you have a well at your home, it can look like a dug well, like you see on the left. Those are about 30 or 50 feet deep. They take water out of sand and gravel. Or it can look like a drilled well on the right. Those go deeper into the bedrock as much as 1,000 feet down. These types of wells need recharge. So if we have a really dry summer, not getting any rain, like we did a couple summers ago, people with more shallow well might have those wells go dry. If everyone in their big family wants to take a long shower, for example, the water might slow down or stop running for a little bit. There is a concept that they talk about nationally a lot called artesian wells, where there's underground pressure pushing water out of the ground constantly, no matter what you do you don't have to pump it, it just comes. These are not common in Rhode Island. The ones we do have, though, I believe are located mostly in Coventry, so you might know someone that has one if you're from the Coventry, Scituate areas on this Zoom. Nationally, though, they come up a lot. But no, we have to pump well out of the ground, not using an old school pump like the one on the right, but by sophisticated systems usually located in the basement. Surface water is connected to groundwater too. So protecting surface water, keeping it clean also helps protect groundwater and vice versa. But groundwater is really important to our state. It's over a quarter of our total water supply. It's the vast majority of water used by farmers for irrigation. About 2/3 of our towns and cities rely in some way on groundwater for their drinking water. A whole lot of the area around URI in Washington County is also on an aquifer, in addition to a lot of the communities in Western Rhode Island. So that is my quick snapshot on these topics, but do your homework and you will learn so much more to complement that. Thank you all for being here today.
Okay, so that leaves me.
We have
I'm just doing a time
questions
check.
for
I-
Molly.
Yeah, why don't we just take a short break, Maureen, and find out if there are any questions before you start?
Happy to answer questions that come up in the future, particularly from schools that took the time to come to this meeting. Maureen, I think we're probably good to go.
Molly, I don't know if you saw, Robin check chat. What is the highest order of streams?
That's some homework for me, Robin, but I'm gonna try and find you an answer. I don't know it off the top of my head.
Sorry. Highest order, we should just like.
Does Situate have a question?
I saw another in the chat.
Yes.
Sorry, I wasn't seeing the chat.
We were kind
Go ahead, Situate.
okay. This might be kind of weird, but we were just wondering if, like, say you had a third order and a fourth order stream that met, and would that form a fifth order stream? Because there was a fourth order stream that merged.
No, that's a really good question, but that would stay a fourth order stream. So whenever two streams meet that are different orders, you keep the number of the highest
Okay.
stream, of the highest order stream. So when a third and a fourth meet, stays a 4. If a fourth and a fifth meet, it stays a 5th.
Okay.
Great question. Another good question I saw in the chat is, are aquifers ever named? And yes, they absolutely are. The aquifer I mentioned that's very important in southern Rhode Island is called the Chepuxet Aquifer. There's also, when we were studying droughts for the theme a couple years ago, there was what was it in Nebraska that I learned about, the Agalola Aquifer or something like that, that serves like a huge swath of the American Midwest. It's very cool. And Natalie, that's a question for Pete and Paul about the national exam. I don't know if that was only shown to us as Envirothon like subject area heads, or if it's broadly available to the teams. That would be up to them.
That was basically shown to the site supervisors, so they understood what questions were asked on a national level. So that when they go to prepare the team for a national competition, they have an idea of the level questions that are occurring on national tests. And really, you only need to can be concerned about the national tests when you win the state and have to prepare for a national competition. We've never released the tests for specifically for the Rhode Island competition.
Thanks, Pete.
Okay.
Supervisors, because then we can borrow from them if we choose to.
Yep.
Oh, no worries, Situate. You don't have your teacher there. I know your teacher. That's why I was so excited to see you pop in. I know Ms. Travers, but you must have been confused, but we're still glad to see you. Have a good one.
Thank you. You too. Bye.
Molly, if you're all set, Maureen, could you please take over?
Thank you.
Sure thing. I know we're technically out of time. Should I like
No, we're
make
not.
this kind of
No, we're not.
fast or just do it normal?
No, you're not out of time.
Okay.
Yeah.
All
Yes,
right.
go ahead.
We booked a whole hour.
Oh, great. Okay, fantastic. All right. Well, then I will jump right in. Hi, everyone. I'm Maureen Dwyer. I'm the education coordinator at the Narragansett Bay National Estuarine Research Reserve on Prudence. Maybe I met some of you out there last year. Just going to share my screen really quick and pull up my presentation. Okay, I'm assuming that's visible for everybody.
Yes, can you switch to a slideshow presentation?
Yep. I just gotta get rid of this stupid bar at the top of my screen so I can find the little, I'll just go to this one. There it is. How's that?
That's
Whoops.
good.
Wah. All right, well, I don't know, maybe you guys aren't seeing it, but my silly Zoom thing is like blocking the whole top of my screen. I wonder how you get rid of that. Sorry. Let me try and.
You should be able to move it.
You just hold it and drag it. Oh, there it is. Yeah, thank you. I'm just gonna bring it down here so it's more out of my way. Okay, great. Sorry for that. I all these years of tech and virtual talks and I still stink at it. Not my forte. Alrighty. So again, my name's Maureen. I work on Prudence Island for the Narragansett Bay Research Reserve. And like Molly said, oh my gosh, there are so many things we could cover in aquatics that it's always kind of overwhelming to try and figure out the best things to share with you guys. But I too took a look at the national test and thought about what we ask on the state test, and just sort of trying to cover these big topical areas. My specialty is more saltwater based wetlands and estuaries and things of that nature. So I'm going to cover wetlands sort of broadly and just get you thinking about different types of wetlands, what sets them apart, why they're important. So most of the presentation is about wetlands and why they are important. And then I have just a couple of slides that talks about ponds versus lakes, what sets them apart. And there's just some vocabulary in there that it might be good to familiarize yourself with. And same thing with aquatic food webs. I don't do much with either of these last two topics, but there's some terminology that would be worthwhile just sort of refreshing your memory with, and it ties back into a little bit about things that Kim and Molly were talking about as well. So with that said, let me see if this will work. Oh, come on. Why isn't this advancing? Oh, there it goes. I'm failing today. Okay, so what are wetlands? Wetlands, and this is the EPA definition. Basically, wetlands are areas where the water covers the soil and the water is present either at or near the surface. And it's also present either all year long or for varying periods of time throughout the year. So it's a little wishy-washy. There are some stipulations in there as to what defines a wetland. So you can have water at the surface, you can have water near the surface, you can have water 12 months of the year, you can have water part of the year. So lots of different definitions for wetlands. So wetlands are found on all continents with the exception of Antarctica. Sadly, here in the United States, at least in the lower 48, we have lost more than half of our wetlands, and that is largely due to wetlands being drained or filled in and built upon either for developmental purposes or agriculture. So, if you think about the city of Boston, the city of Providence, those were all once wetlands and now they're giant metropolises. So we've lost an awful lot of our wetlands, and with those go the various species of plants and animals that they support and all the ecosystem services and benefits that wetlands provide us. So wetlands are vastly important. They're found throughout the world, essentially. And as we're going to learn in a little bit, they serve a lot of really important functions that we would normally get for free from them. But when we fill them in and drain them and build over them, we lose all of those valuable ecosystem services. So more to come on that in a bit. These are terms that are just really good to know. So take some time to learn these when it comes to test time, especially. So hydrology, hydrophytes, and hydric soil. So 3 H's, that'll hopefully help you remember them. So these 3 things are kind of what makes a wetland, if you will. So hydrology is simply the amount, distribution, and the movement of the water in a given area. Hydrophytes are those types of plants that are specially adapted to growing in wet soils or saturated soils. So wetland plants are hydrophytes. And then hydric soils are simply soils that have that really long period of saturation. And this often results in very low oxygen conditions. Not quite anaerobic where there's no oxygen necessarily, but low oxygen conditions. So once again, just kind of know those terms: hydrology, hydrophytes, and hydric soils. In terms of wetlands and what they are, or what types there are two main, at least I'm breaking them down into two main types. We have the inland or the non-tidal wetlands. So those are ones like freshwater marshes, bogs, swamps, fens, and vernal pools. I think the national competition is in New York this year. So I would, if you make it to nationals, we would want to think about what kinds of wetlands are specific to that area, and make sure you're familiar with those. So those are the inland non-tidal wetlands. And then there's also the coastal or tidal wetlands that include salt marshes, which Rhode Island has plenty of, and also mangrove swamps, which you have to go all the way down to Florida before you start running into mangrove swamps. They're a southern species, mangroves are. So I'm just going to touch upon a few of these real quick and sort of tell you what sets them apart. So swamps dominated by trees. That's the thing to remember about swamps, as opposed to something like grasses, which dominate salt marshes. Swamps have water present year round. The land is permanently filled with water. The soils have really poor drainage, and often they'll be specifically called what type of swamp it is based on the trees that grow there. So if you go down to the mid-Atlantic or the Carolinas, and further south, you might experience what's called a bald cypress swamp. And that's simply because all those trees in that photo is dominated by bald cypress trees. They have the really wide buttressed roots. It helps stabilize them in that habitat. They're beautiful and amazing species. And then again, likewise, you could have a hardwood swamp, and that might be filled with maples and other variety, sweet gums and tupelos, other varieties of hardwood trees. And swamps is kind of, when you think about swamps, you think about creepy crawly snakes and alligators. And that's because swamps are really important to a lot of different species of reptiles and amphibians. So snakes, turtles, gators, reptiles, amphibians, all that are typically found in swampy systems. But they're really an amazing ecosystem, amazing habitat, and provide just a great habitat for a diversity of wildlife. Freshwater marshes, on the other hand, dominated by grasses, cattails, rushes, reeds, a little more open. You probably have the edges is maple trees and things of that nature. But when I think of a freshwater marsh, I always think of oh, that's a place where the beavers have probably chopped down a bunch of surrounding trees and created a dam and built a beaver lodge. That's more of your typical freshwater marsh. And this is a photo that I took in Connecticut of a freshwater marsh over there. Vernal pools, these are more, these have water on a more seasonal basis. So these guys tend to have water in the winter and then in the spring, and by summer and fall, they've gone dry. You're probably familiar with vernal pools as being super important habitat for many different species of reptiles and amphibians to lay their eggs. So by laying their eggs in a body of water that is temporary, that body of water is not going to be home to things like fish, which would be a major predator on things like the eggs or the little juvenile tadpoles. So vernal pools are very, very important to the survival of many reptile and amphibian species. And just know that they are generally temporary bodies of water, drying up in the summer. Bogs versus fen. These are pretty cool habitats. Bogs, even locally around here, support really cool plants like pitcher plants, little carnivorous type plants that you don't really find anywhere else. The big difference between bog and fen is that bogs only get their water from rainfall or snowfall. So it's just strictly from precipitation, whereas fens can receive a big portion of their water and nutrients from groundwater sources. Obviously, precipitation will help a fen maintain water as well, but fens also will get that water coming up from the ground. So know the difference between how bogs and fens receive their water. Switching gears and thinking about the coastal or tidal wetlands. So I'm not going to talk about mangroves because we don't have any up here, and the national meeting is not down in the deep south. So we probably don't need to worry about mangroves too much. But salt marshes are, thankfully, they are pretty abundant in Rhode Island. So if you've gone to the coast, you've probably seen a salt marsh. They're up and down the East Coast. They are influenced by tides. So you have low tide and high tide, which means anything that is living in a salt marsh, plant or animal, especially if you don't have the ability to fly away like a bird. You need to be adapted to some really significantly changing water quality parameters or water parameters. So you've got flooding at high tide, totally drying out at low tide. If it's the heat of the summer, you've got 95-degree water or baking hot sun. And then in the winter, you've got ice maybe covering everything and, below freezing temperatures. You have varying amounts of salinity. If it's pouring rain and it's low tide, your salinity might be very low. And versus, an incoming high tide from the ocean, it might be very high. So animals and plants that live in salt marshes generally can tolerate a wide range of all of these different parameters of the water, particularly dissolved oxygen, temperature, and salinity. And just in terms of ecosystem services, salt marshes are all wetlands serve many, many functions. So actually, I'm going to slip ahead to this next slide. So wetlands, just so many different functions and values of wetlands, everything from providing recreation areas and fun for humans to food source and habitat for different types of wildlife. A lot of the food and species that live in the salt marsh and in estuaries are things we like to eat, right? So it is providing people with jobs who go out and catch clams and oysters and fish and crabs. And it's giving us the ability to go to a restaurant and order that and eat it. So not only is it food for wildlife itself, but it's food for us humans, and that turns into jobs, and it turns into the economy and being valuable for that. Wetlands are also great at helping to reduce flooding. So salt marshes in particular, marshes are like giant sponges. So they have the ability to absorb excess water. And so if we get a nor'easter or a hurricane or just really strong onshore winds with a super high tide, the salt marsh has the ability to act like a sponge and soak up a lot of that extra water. And that's going to limit the amount of flooding that happens on the mainland, which might otherwise flood a very highly populated place or damage infrastructure, and that can cost a lot of money to repair. So marshes can help to mitigate that and buffer that. Marshes and wetlands in general are great at cleaning the water. They're kind of like their own little water treatment facilities, if you will. And I think my next slide shows that a little bit. So imagine, you have a high rainstorm. The water runs over roads and city streets and impervious surfaces, picking up lots of nutrients, pollutants, fertilizers, pesticides, waste. All of that stuff comes running down into a low point. If you think about watershed at this point, all that water is running down into a river or a stream, which then makes its way into the bay. Well, hopefully somewhere along that line, that water is flowing over a wetland. And then that wetland will have the ability to slow that water down, filter some of those toxins and pollutants and bacteria out of the water, so that by the time the water leaves that wetland, say like a salt marsh, and that water makes its way through the marsh and enters Narragansett Bay and then the ocean, that water is much cleaner upon leaving the wetland than it was when it entered the wetland. Of course, there are human impacts on wetlands. There are plenty of them to mention. I've listed some of them out there. They're pretty obvious. They're probably ones you're already thinking about. So again, we have filled them in, we've ditched them, we've drained them, we've built on them. We might cut a channel through it and alter the way the water moves naturally. Just simply by having a high density population, we increase the level of pollutants. There's more maybe oil being dripped from cars. There's more fertilizers being put on lawns or on golf courses. There are pesticides being placed in agricultural lands or on people's own properties. And then, of course, the introduction of non-native species, which often can outcompete our native species. And that just throws the whole the whole ecological balance out of whack, if you will. And it can really change species composition. So invasive species are the number two threat to the loss of biodiversity in general, not just in wetlands. The number one threat being habitat loss, but number two is invasive species. So they are really, really impactful in terms of loss of biodiversity. Okay, so just the last few slides. So a pond versus a lake. We don't often talk about this too much, but I think it's worth noting. So ponds generally are shallower, they're smaller, and there is no aphotic zone. So the aphotic zone is absent, which means there's nowhere in that body of water that light doesn't reach. So light can get all the way to the bottom in a pond because it's that shallow, versus a lake, which generally are bigger, deeper. The lakes will have an aphotic zone. So at the bottom of the lake, there may be little or no sunlight that is able to reach that depth. And then lakes also generally have a stream feeding into it or draining from it. And then in terms of succession, over time, and we're talking probably centuries of time here or more, lakes will slowly fill in with sediment. They'll eventually become a pond. The pond will continue to fill in. Those cattails, those reeds, those grasses start to grow in thicker and thicker, and then eventually it'll become a forested land. So you go from a lake, it starts to fill in, it becomes a pond, it fills in even more, and will become a forested land. And again, talking like not in my lifetime, like it's probably many centuries for that to occur. One thing I've noticed some terminology coming up in some of the national tests are things like oligotrophic. So I just wanted to throw this slide up here so you could familiarize yourself with this terminology. So the trophic state index just tells you basically the quality of the the quality of the water in the lakes. There's a term for it, and I had it written down, and I don't have it in front of me of what it exactly means. But oligotrophic is basically excellent water quality, very cold water. The oxygen is present all the way at the bottom of the lake year round. There's high dissolved oxygen, which means it supports fish that require very high amounts of dissolved oxygen in the water. It's clear water, it's cold water. So oligotrophic is excellent water quality in a lake. Mesotrophic is somewhat clear, but there's a better chance that you have very low dissolved oxygen in the deeper parts of the lake during the summer months. So remember, I think it was Kim that mentioned warmer water holds less oxygen. So we would expect that to be the case in a mesotrophic system, where in the summer months, the water's a little warmer. So even at the bottom of the lake, you might have low DO. And then eutrophic is when, if you've studied eutrophication at all, a eutrophic lake is dominated by blue-green algae. You have plant scum kind of everywhere. Excessive plant overgrowth problems are possible at this point. So just throwing those words out there to kind of put them in your brain. And then one last thing to touch on is aquatic food web and energy flow. This is pretty basic if you've studied food webs at all. So just thinking about it in terms of aquatic systems of what that might look like. So you might have cattails or grasses or algae as your primary producers, right? And then you have your primary consumer, secondary consumer, tertiary consumer, so on and so forth. So I created one from critters that I have seen out on Prudence Island, just to give you an example of how trophic levels work and the energy transfer within them. So your salt marsh grass is your primary producer, and then you've got things like phytoplankton, zooplankton, and then mussels eat the plankton, and then a blue crab might eat a mussel, and then the sea robin eats the blue crab, and then the osprey eats the sea robin, so on and so forth. But what's important about this slide is to remember that only about 10% of energy is available to the organism at the next level in order to make itself new biomass. The other 90% is basically lost as heat and undigested materials. So just know that when they're talking trophic levels, only 10% of energy is available to the organism at the next level. So it gets kind of smaller and smaller as you go up that trophic scale. So hopefully that makes sense. There's some really great websites out there on trophic levels. There's some really great diagrams. So it might just be something worth reviewing. And that's it. I'm done. I hope I didn't go too long. I will stop sharing.
No, I don't think so.
Okay.
Any questions, anybody that's still here? Situate had to leave.
When I practiced this, I had all my little, it like showed me my screen with all my notes on it that I was going to say on each slide. But when you screen share, that totally goes away. So some of my terminology that I had just learned, because I don't know much about like trophic lakes and all that jazz, I did not have in front of me. So I apologize for not knowing quite
You
as
mean
much
you're
as
almost like the students, you have to learn stuff?
Yeah, I do.
Just
Hey, if you don't teach it, you don't learn it. I mean, if I don't teach it regularly, it's like, gone. So yes, we're all still learning on a daily basis.
Robin,
just-
anything from you guys?
No, I think we're starting to trickle out. There's 5 of us left in the
I
room.
did,
But that
Okay.
was great. Thank you all very
Thank
much.
you for coming. It's nice to have some
Excellent.
people in the room.
Yes,
Right,
thank
right.
you very much.
I did want
And-
to throw in there real quick on top of the terminology that Maureen gave you. One part of the presentation I did not get to was the difference between lentic and low-tick systems, whether lentic being like your lakes, ponds, very slow moving, non-moving water, whereas low-tick is flowing water, rivers and streams. And I think that's pretty important because I know that's popped up on the
One of the things also
national test a
that
couple
we may
times.
want
So
to look at is the types of fish that are in New York State.
Yes.
Yeah.
And
Absolutely.
invasives.
Yes.
and invasives
Yep.
is huge.
Yeah.
'Cause It is,
So-
it is the Finger Lakes area where we're gonna be, Geneva.
Cool.
Yeah, so we'll figure that out and what is up there and stuff like that.
And we'll see you in April, Kim.
Absolutely, April 27th, right?
Yep, we're coming down to
Okay.
the Campy hunting.
Yay.
Cool. Okay,
Awesome.
hey
I'm gonna
Natalie,
sign off then.
all right, Natalie,
then,
stay on for a minute.
No
The
problem.
board meeting will be on Thursday up in the Northern District.
Yep,
Because Eastern Bay is in trouble.
Yeah, what a mess. I
Yeah.
am so glad I don't have to be on that road.
all of a sudden the bridge
No.
just crumbles. It's like, wait a minute, how many
Holy-
years has that bridge been there?
Yeah,
Yeah,
exactly.
right?
Cool,
But have to start
Okay.
taking the ferries again.
Yeah.
there we go. Or
Oh,
take
that
the
was-
train.
Yeah.
Welcome to my world.
Okay,
Thank you very much, everyone, and thank you, ladies, very much for your presentations.
yep,
You're
enjoy the
welcome.
day.
Thank you.
Thanks for having us. Nice
Okay.
to see
All
you
set.
all.
Peter, you can stop recording, and we'll talk to you on Thursday.